Our first speaker today, a little interesting story here. We were thinking about who to invite as speakers at DAS. And I had actually been engaged in a little process, a little research process. I was working for the integrator Chesapeake Systems at the time, and was doing a little research that was beckoned by a long friend of mine who's a genetic engineer. And he said, you know, Nick, have you ever heard of storing data in DNA? I said, well, I have heard of storing digital information in DNA, but that's like the kind of thing that'll happen in, like, you know, Star Trek world. You know, it's like the ideal data storage. But he said, no, you should look at this. And I started researching it, and very quickly a Boston-based company called Catalog popped up, and I engaged with their CEO, Hyunjun Park, and I said, you should speak at this conference that we have called DAS, because this is the perfect subject. It's a bunch of media archivists, preservationists who deal with digital issues, and you know, the lack of archival mediums for digital is huge. Please speak at it. And here I am today, and I actually work for Catalog now. This is a shift from last year. I took a job with them. This is, I think, my 7th week now. And it was such a fascinating subject and something I felt so passionate and so connected to. I thought this is the only thing that will probably get me to move on from Chesapeake ever. So I took that opportunity. So we're actually going to start off, before Hyunjun comes up and speaks, we're actually going to play a 10-minute video that Catalog produced last year to give a little bit of an intro to their company's activities. And then Hyunjun will come up and give his presentation. So if you guys want to play the Catalog video, we'll get started with that. And again, thanks everyone so much for joining us today.
So the photo is in that too?
Yes.
Wow. Okay, hang on. First, let's start with a quick story. This is a self-portrait of Sergei Prokudin-Gorsky, one of the earliest pioneers of color photographs. The Tsar of Russia was so blown away by Gorsky's 3-color method that he gave him a train car to use as a portable darkroom and sent him out to take color photographs all across Russia. He traveled around like this for about 6 years, taking thousands of incredible photographs of Russia during what turned out to be the eve of World War I and the Russian Revolution. Gorsky fled Russia, and the only reason these images survived is because they were rushed into a box and spirited across Europe to some basement in Paris. Now, they represent one of our only glimpses into this incredible world of the early 1900s Russian Empire. About half of his photo negatives were confiscated by the Russian government before he could leave and were probably destroyed. We'll never know what the other pictures looked like. They were swallowed up by the chaos of an uncertain and rapidly changing era. These days, we're all Gorsky, documenting our lives and our world obsessively, creatively, but much more casually. When we take a picture, record a video, or send an email, do you really expect it to last anywhere close to 100 years, like Gorsky's delicate glass slides? The cloud is just someone else's computer taking up acres of land, consuming cities' worth of power, and billions of dollars in cost of ownership. And it ties the fate of our data with the economic fate and privacy policies of a provider, not to mention generating more carbon emissions every year than the entire airline industry. And as you might have noticed, we're in the middle of an explosion of data creation. The amount of data that we produce each year as a species is growing exponentially, while our storage capacity is not. Hard drives are not going to keep getting smaller. We've basically reached the physical limits of that. Data centers and data storage warehouses are just going to have to keep getting bigger and bigger and bigger and just multiplying across the face of the earth in order to keep up with the amount of data that we are generating. On top of all the space, energy, and money that our data is sucking up, there are some serious concerns about the integrity of our archival materials in the long term. Hard drives need to be replaced about once every 5 years, and magnetic tape can last maybe 30. So do you really think that your data is going to last for posterity if it's stored on stuff that goes bad so frequently and goes out of date even more often? Think 100 years down the line. What about 500? What about even 50? All this stuff put together has created this looming slow motion crisis. We kind of need to figure this out. And it turns out the answer has been with us since the very beginning. Literally.
DNA is the medium that nature has chosen to store its most precious data. What is data? That's a really hard question. For the context of digital data storage, it's just any information that can be reduced down to a series of ones and zeros. But when you think about what is information, recording information and keeping it for posterity is what made civilization possible, what brought us out of prehistoric times and made the accumulation of knowledge possible so that we can evolve beyond just one generation's worth of knowledge. My name is Hyunjun Park. I'm the co-founder and CEO of Catalog. We're an early-stage startup focusing on storing information with DNA molecules.
You know, there was never really a eureka moment, but you know, it's something that developed organically over the courses of weeks. It kind of started out with the idea that other people that are approaching this problem are approaching it from a standpoint that's very expensive and actually time-consuming when it comes to storing digital information.
We came up with a way to encode information in DNA molecules in a much faster and cheaper way, and so we decided to take a stab at it.
As with everything else engineered by humans, we gain inspiration from nature. If we see how the birds are flying, we will think of ourselves flying with airplanes, and the same with DNA. We see that our bodies store information on it, so why not store data on DNA?
DNA has a lot of properties that make it very attractive for data storage.
The information density of the material is 1,000,000 times higher compared to things like flash drives. So that's up to an exabyte of data that you can store in a sugar cube's worth of DNA. And an exabyte is about the information you can store in a large data center. Usually a data center is about the size of a football field. So you could store all the information in a large data center if you were to do it in DNA and the size of a sugar cube.
And then another thing, it lasts for such a long time. It's really, really stable, and you can pull information out thousands and thousands of years later.
DNA can last for potentially thousands or hundreds of thousands of years without degrading or being corrupted. So this is the reason actually that we know the sequence of the woolly mammoth, even though it went extinct 60,000 years ago.
The idea actually of storing information using DNA molecules has been around for several decades. So people have been looking at how nature stores information in DNA. So it's the linear sequence of the base pairs that map onto the sequence of proteins. So people have been trying to do it backwards. So you map the series of ones and zeros, the binary code that you want to store, onto the series of base pairs of DNA and synthesize enough DNA to store all of those numbers. It might be an intuitive way to go about it, but it's very expensive and slow to do it that way.
The technique that we landed on for storing information in DNA involves synthesizing a bunch of prefabricated DNA. So you can think of the prefabricated DNA as an alphabet.
It's like going from copying down a book using a pen and paper to now using a printing press with movable typefaces.
So first you have your picture, and you have it as a digital file, so maybe it's a JPEG.
At that point, we turn it into a string of binary. We break it up into different blocks. The information gets translated into a pool of words, which are a pool of DNA sequences that represent it.
So then we have our building blocks in a plastic plate in a bunch of different wells, and the robot is able to pick from those different wells and combine the right pieces, the right building blocks together. And then the next thing we add is an enzyme. And this is a biological molecule that then takes the pieces of DNA and sticks them together.
So I would form a molecule, and this molecule will encode a piece of information, a piece of the picture.
And then on the robot, we can transfer each of those assembled identifiers and put them all in one big pool. And now you have all of your data stored in one tiny little tube.
So the photo is in that too.
Yes.
Once you have all of the DNA written, it just looks like a translucent droplet, but it actually contains millions, if not billions, if not trillions of copies of your information.
If down the road you want to look at your data again, you can take your DNA out of storage and put it through a sequencing machine, which just reads back the DNA molecules.
So we kind of have a dictionary of sorts for DNA, where every word has an inherent order to it.
Then a computer can read it right back into a JPEG from the binary.
What we're doing, kind of the standard of what we're doing, is super exciting. The idea of storing terabytes and petabytes of information in DNA is more than enough to get me out of bed every morning and into work. But I think what really gets me excited, thinking 10, 15 years down the road, is what the implications are once all the data is stored in DNA.
I'm actually really excited about things that we just can't even think about now. So to transport a lot of information right now, for example, the most efficient way to do it would be to ship hard drives rather than to send it over the internet, because internet bandwidth is too slow. So DNA could actually become the most efficient way to transport information.
Put a droplet on a piece of paper and mail that to someone if you wanted to.
I really like the idea that we could have like a library of human civilization in the Arctic in a refrigerator.
What we think is we discovered a method that can revolutionize the way how people store data.
Decentralized data storage is becoming a really popular concept now as people are realizing that most of the data that you store on the cloud is actually stored by 2 or 3 companies on the back end. So if any change in power were to occur, or if any problems were to occur within these companies, all of your data would pretty much be at risk.
But DNA-based storage could change all of that. Everything you ever generate can be stored in a very small pellet of DNA. So with that, you could have physical ownership and control over how your data is stored. So with the machine that we use now, we can do very small scale demos and megabit scale demos. So we're currently building a machine that will get us to a terabit per day of writings capacity.
That's a pretty rapid growth.
Right, that's a million-fold improvement in the capacity. But we've been working on it for the better part of last year, and there are a lot of very motivated people working on the project.
I love building cool things. I love building systems that nobody else has even thought of before. This is definitely one of those things, but an even stronger motivation to me is that the thing we're actually building could have really powerful implications for the future of the world, really. So, you know, this data storage problem, people typically don't think about it, whether or not their data will be around 30, 40, 50 years from now, whether their data that they have today will be accessible by their children or their grandchildren. A lot of us really tend to live in the moment, and you know, it's possible that towards the end of our lives, we're all going to be looking back and wishing we had saved some of those moments that we created when we were younger.
Logistics. Hold on. Ta-da.
Good morning. My name is Hyunjun Park, co-founder and CEO of Catalog. I thought I'd start out today with a brief history of information storage in DNA. So, it's a very brief history. But the inherent characteristics of nucleic acids that make them the perfect choice for information storage, as well as computation, for nature, is a result of billions of years of evolution. The selective pressure to put all of that information, pack all of that information into a microscopic cell, demanded incredible information density. And as you can imagine, the extreme chemical stability as well as the ability to replicate that extremely fast were things that were highly selected for. 3 billion years ago, there was a small upgrade from RNA to DNA, but for the better part of 3 billion years, information has been stored and computation has been done directly on those molecules, on the molecules of DNA. Now, even before the discovery of the structure and mechanism of DNA, physicist Ernst Schrödinger argued that life could be explained and thought of in terms of storing and passing on biological information. He proposed that because so much information has to be packed into such a small cell, there has to be a hereditary code script embedded in the molecular fabric of chromosomes. To understand life, then, we would have to identify these molecules and crack their code. Now, around the same time, Oswald Avery showed that DNA and not protein was the genetic material. And finally, in 1952, Rosalind Franklin, her seminal work on the X-ray crystallography of DNA led to the discovery of its structure. Now, to this point, I've been talking about biological DNA. So that's the stuff that's found in cells, and that's composed of the A's, G's, T's, and C's that you're all very familiar with. Inside of the living cells, the cellular machinery comes in to transcribe the DNA molecules into RNA, and that gets translated into protein, and the linear sequence of the base pairs of DNA gets translated ultimately into the linear sequence of the amino acids of proteins, which make up your body. But when I talk about storing digital data in DNA, I'm talking about synthetic DNA. So it's still composed of the same material, the same A's, G's, and T's, and C's, but they're not isolated from biological cells. Instead, they were chemically synthesized from the components, component nucleic acids, and the end result is synthetic DNA molecules that have the same characteristics as biological DNA, so that they can be manipulated using the tools of molecular biology and read back using the exact same machines that you would use to sequence genomic DNA. And after synthesis, these molecules will be stored inside of a laboratory test tube, so they're not usually put back into cells. There's no need for that. And they're very happy as a dried down powder in the bottom of the tube at room temperature. Now going back to the history, as early on as 1964, the possibility of storing digital information, arbitrary digital information in DNA was considered. The Soviet physicist Mikhail Nieman is credited as the first person to suggest the idea of recording, storing, and retrieving digital information at a molecular level, including the use of DNA. So he suggested other methods as well. The first one to actually do it, as far as I can find, is an artist named Joe Davis back in 1988. He collaborated with scientists at the Harvard Medical School to encode this symbol of fertility and the female Earth into DNA. So it was a very literal depiction of that icon, and that sequence was put into DNA. But this time, the synthetic piece of DNA that was synthesized was put back into a cell so that he could create a living art. Excuse me. Now, after that, the field has been kind of quiet for a long time, until very recently, possibly driven by the Human Genome Project. The sequencing costs of DNA has been falling very precipitously. So you'll notice that the graph is dropping exponentially on already a log scale. So Moore's law is already an exponential trend, and the sequencing cost of DNA is dropping far faster than that. So from that, in 2012 and 2013, back-to-back papers came out of the labs of George Church at Harvard Medical School and Nick Goldman at the European Bioinformatics Institute, which were really the first instances where a significant amount of data was put into DNA. So this was the first work where they both kind of encoded about 5 megabytes of data into DNA using next-generation synthesis and verified using next-generation sequencing methods. In 2016, Microsoft announced the encoding of about 200 megabytes of information in DNA. And this work is still holding the most amount of data that's been stored in DNA molecules. But the MIT Tech Review that wasn't involved with that research firsthand estimated that it would have cost them about $800,000 worth of DNA synthesis to generate that much DNA. In terms of information density, that record belongs to Yannick Ehrlich, who was then at Columbia University, and they were able to encode information at a density of 214 petabytes per gram of DNA. So that's about this much DNA. And most recently, Microsoft published their recent work, their latest progress in the field. The focus of this one was fully automating the entire process from end to end. So you give the system a message or data, it encodes it and puts it into DNA, stores it, and then retrieves it, sequences it, and gives you back the original message when you request it. So that's it's great progress for the field, but the focus was not on the throughput or the cost of DNA synthesis. So the message that they were able to encode was hello. Now, it's not just the dramatic decrease in sequencing costs that is really driving this field. As you all know, it's we're living inside of an explosion of data creation. And we try to solve that problem by sending things off to the cloud, but since there's really nothing special about the cloud, we'll only be able to store a small fraction of the useful data that we'll generate. Not only that, the internet isn't the most efficient way to transport massive amounts of information, and even after we've stored everything, in order for data to add maximum value, we want to actively use it. So that's also driving not just a shortage in storage, but also in computation. Our conventional clusters are currently limited by the speed at which we can remove heat away from it, because you can only move electrons around so fast before it heats up too much. And so that's why we're turning it to DNA. It's again the medium that's been selected by nature and has evolved over billions of years. It has the ability to store an entire data center in the volume of a sugar cube, and to retain that data for thousands of years without migration, and to make millions of copies of that for free, nearly for free. And as long as we remain carbon-based, we'll always be able to read back DNA. And finally, we're all examples of massively parallel DNA-based computers, and we can harness that power for things like inference across an exabyte database. And there's some classes of problems that are perfectly suited for DNA-based computing. Those are data parallel problems that go past the practical limits of any clusters of processors. This is because with DNA, the computation happens in the very molecules that the data is stored in. Excuse me. Each molecule of DNA that stores the bits are in themselves coprocessors. So the more data that you store, the more coprocessors you're adding in a one-to-one manner. That's very different from an I/O-bound computational scheme where no matter the specific architecture, the moment the data that you're computing on makes it necessary for processors or clusters of them to talk to each other, then you're limited by the amount of heat you can remove. And even with parallel processing in the conventional sense and other enhancements in technology coming into play, the reduction in runtime will really largely be negated by the overwhelming increase in data. In comparison with DNA, you can truly achieve a scale-free computing because you're adding coprocessors in a one-to-one manner with the data that you're storing. So that's all great, and where are we with this technology? Why hasn't DNA taken over already as a mainstream storage and computational medium? Traditionally, so you've seen it in the video, but I'll go over it very briefly again. Researchers in the field have been very focused on how nature stores information in DNA. So the linear sequence of the base pairs of DNA, the A's, T's, G's, and C's, ultimately maps onto the linear sequence of the amino acids of proteins. And so studying that, it's very intuitive to try to store information, digital information, in the exact same way, but in reverse. So if you were trying to encode the data that you see on the bottom, the ones and zeros, you would define a linear mapping between the bits and the sequence of the base pairs of DNA and synthesize enough DNA to store all of that information, to represent that using the DNA base pairs. But again, this is a very slow and expensive process. So we moved away from that and decided to forget everything temporarily that we know about the central dogma and just thought about DNA as a nanoscale polymer that we can predictably manipulate. And doing that made us realize that the cheapest and the fastest way to generate massive entropy using this material isn't to synthesize new DNA de novo, but to combine and assemble pre-made DNA molecules to create unique sequences. it's the DNA equivalent of going from a pen and paper to now using a printing press with movable typefaces. And that allows us, it's a simple idea, but that allows us to achieve about 100,000-fold improvement at least in speed and cost. And so that was picked up by the media, hailed as sort of the beginning of an ecosystem where DNA is the central player in data storage and computation. And I alluded to it in the video, but over the last 2 years, we've been building a machine that implements our platform at a massive scale, so terabit scales. And as of yesterday, we successfully completed a run where we encoded gigabyte levels of data, and it didn't cost us hundreds of millions of dollars to do it. And so stay tuned for the full press release in the coming days. But it's a machine, schematic is shown here. So it's a sizable machine, takes up the better part of a large room, but it's getting us to a throughput that's never been possible before at cost points that's also about 1 million times cheaper than what's been possible before. So with the completion of the machine, we're in the process of signing up visionary customers across many different industry sectors. And Nick, since joining 7 weeks ago, has been hard at work speaking with many of you in the room. And we look forward to building this technology with you. So I spoke about all the potential DNA has in data storage and computation, but we don't think that that's the limit here. So imagine a world where you can store all of your medical information and carry it with you on the person in a very accessible way. That way, it's totally offline, so you don't have to worry about it getting hacked or having other people getting unauthorized access to it. But it'll always be accessible to you because all hospitals have sequencers. And we also talked about distributed storage across thousands of locations for things like blockchain ledgers. And if we're ever going to colonize Mars, shouldn't we send people off with the entire accumulated knowledge of the human race? We can only do that using a medium that has the information density of DNA. And through the years, others have also come up with some creative uses for this, around this theme. So the first one on the top right this group used synthetic DNA sequences to encode a computer virus, which was then uploaded when that molecule was sequenced and processed inside a computer. Next one, some people with millions of dollars in cryptocurrency decided to put their passwords into DNA molecules. We've seen examples of people putting entire albums at a very high cost into DNA. We've seen it being used to track the flow of oil under the ground. And where also a case where a GIF was put into synthetic DNA molecules, put back into a cell, and the CRISPR system was used to integrate that synthetic DNA into the genome of the organism. My personal favorite, in the 70s, researchers wondered if a bacteriophage, Phi Chi 174, if the genome of that contained messages from intelligent life elsewhere in space. And so this was actually the very first genome of a biological entity that was sequenced. And bacteriophages, to back up, are small viruses that infect bacteria but are safe to humans. And so they have very small genomes, and it was the very first genome to be ever sequenced back in 1977. So when they had this full sequence, they wondered, could this have a message from aliens? And they were very inspired by Carl Sagan and Frank Drake's thought that intelligent civilizations may try to communicate with us through a two-dimensional image. And so when they looked at the genome and tried to decode any possible messages that were in there, they noticed that 2 of the genes were overlapping by 121 amino acids. So an amino acid is encoded by 3 base pairs of DNA. And so this is the region of the genome that's overlapped for the 2 genes. And this is where it gets really brilliant. They noticed that 121 is a square of 11. So they decided to hash that into an 11 by 11 matrix. And in each of these cases, either they took the first letter of each of the codons and put it into a matrix, or the second letter of each of the codons, or the third letter of each of the codons. And then depending on the chemical composition of the base pairs, they decided whether to make it black or white. Unfortunately, they couldn't decode any kind of alien message through this effort, and they kind of stopped there. But that's very unfortunate, because now we know that if they had only thought to arrange those in a circle rather than a square, they may have had better luck. Great. So that's what I have prepared for you today, and I'd be happy to answer some questions for you.
All right, I'm going to be directing a couple of our volunteers around the audience. If anyone has questions about DNA data storage. We have, I think, a question in the back, right there, a waving hand.
Hi, I actually have a million questions, but I'm going to try to ask two. So first of all, when you say, okay, so computer is a binary, zeros and ones, DNA has 4 factors. When you're mapping one to the other, is it that a 0, 0 becomes a G, or is it that the 0, 0 goes directly onto the DNA? Is that a weird question to ask? I'm basically asking, is it translation, or is, oh, sorry, I missed this slide initially. And then my second question is there an effort to be made to, instead of the translation process, it seems like that is quite a significant amount of what your computer, like your current machine's effort is going to be doing, is this translation, right? This mapping from one to the other. Is there a world where you envision the data being created in the AGTC code, like it created as DNA at birth?
Well, one thing I'll say, this isn't how catalog encodes the data, right? So maybe you can speak a little bit more. It's highly proprietary information. We'll have to kill all of you if we tell you all the secrets. But maybe you can speak a little bit to that kind of breakthrough in that realization of how the encoding scheme didn't need to be as sort of arbitrary as direct mappings of ones and zeros. And then the follow-up is, you know, we've been thinking in terms of 2 numbers because that's a very easy thing to encode digital information, which anything can be reduced down to. But now that we have a medium that is capable of capturing thing in 4 units, essentially, has there been any thought of, you know, what, you know, would that be quaternary data would look like, not binary data, and whether there would be any utility to kind of natively generating digital information in that quaternary fashion?
Yeah, I think to try to answer that second part first, I don't think the field has gone on to really trying to develop a new way to translate life or the world into a different language. You know, I think it's been more about taking what's already been developed in this digital world of binary code and trying to represent that in the best way possible using DNA as just a medium. And I think you were asking about whether we use the actual ones and zeros or the base pairs, but anytime you're using a medium for data storage, whether it's hard drives, flash drives, or DNA, you're essentially just representing the ones and zeros in a very conceptual way. So, right, even with magnetic tape or hard drives, you don't actually write in 1 and 0 in that medium. It's maybe a magnetic polarity or the presence or absence of a charge that conceptually maps onto the ones and zeros. And the same thing is done with the traditional way of storing information using DNA. So the ones and zeros are conceptually represented using a quaternary system. So a base 2 system mapped onto a base 4 system, meaning theoretically each of the base pairs can represent up to 2 bits. And so that's what's been done before. We found that information storage is just about representing ones and zeros, and that's a matter of entropy. You don't have to represent it in this way where it requires a lot of DNA synthesis, which is hard, expensive, and slow. So what we do instead, very briefly, and I can explain that in more detail offline, is we have the ability to generate lots and lots of very different, unique molecules that we can tell apart from each other, but starting from a set of pre-made molecules. And what we do with these molecules that we've generated that have trillions of different unique molecules in them is have each of these molecules represent a unique bit within the bit stream. And I think this may be going a little bit too far into the weeds, but each time there's a 1, we make the molecule that represents that cell. Each time there's a 0, we choose not to make it, so that the collection of molecules are, when we pull them together, is just the collection of 1s where the order is known, because we have a lexicon for it.
All right. Any other questions? I have one. Well, first we'll get to you, and then I'll ask mine afterwards. Thank you.
Hi, thank you. So you mentioned the artist Joe Davis and the symbol that he created and injected back, or created with, synthesized into DNA and injected back into a cell. You also mentioned the dry powder of the storage system at Catalog, which is not back in the cell. Could you just talk a little bit about that, because here's art going back into a living component, storage not, but science fiction has other ideas.
Right. I think the motivation behind Joe Davis's work, where he put that piece of DNA into a circular piece of DNA that's easier to put into cells just because of the chemical qualities there, it was more driven by his artistic motivations rather than to preserve that information in a very high fidelity way for posterity. Because when DNA is inside of a cell and it's actively replicating, mutations can be introduced. And there are other events within the cells that can either eat up that foreign piece of DNA, CRISPR being one of the innate immune systems of bacteria that can be utilized to do this. Or there could be a myriad of other things that could go wrong with that. But when we're looking at synthetic DNA and in the context of data storage and computation, we want it to last forever without ever having the base pairs changed. So we don't put it back into a cell. There's really no reason to do that. And when we manipulate it, it's in solution, it's a water solution. And when we pool it all together and keep it for storage, we would freeze dry it essentially. So it's just like a salty powder in the bottom of the tube. And that's the condition that we preserve it in. And in that condition, it's much more stable than any other traditional media. So when I talk about DNA, people often ask me about how stable is it against things like extreme radiation. Well, I think that's missing the point a little bit, because you wouldn't subject your hard drives or flash drives to extreme radiation. So it's at least as stable as the traditional media. But since it's the presence or absence of physical molecules that encode your information, it's much more stable over long periods of time.
I'm going to interject my own question, not that I don't know the answer, but I thought maybe you could elucidate this for the audience. You spoke to the terabit per day writer, and so that's about 125 gigabytes of data that this combinatorial approach can get into synthetic DNA that's been prefabbed and combined and enzymatically stitched together. And that's great. That's many more times of data than some of those previous efforts you spoke to. We can get movie files in there. We can start to get some interesting stuff. But what do you see a few years out? What starts to turn the fact that we have this medium that in very microscopic, tiny exabyte and sugar cube sized volumes can store limitless information, but obviously there's that bottleneck of writing to it. Where do you see the evolution of the technology going over the next few years to start to potentially ramp that up?
Right. So I think I'll have to speak to sort of the development timeline and goals of the company. And because we're an early stage startup that's venture backed, we work in these increments. So we raise our seed round, and without the money that we've raised, the amount we could get to that we could foresee was the terabit per day. And because this was a totally new embodiment, even though we were taking components off the shelf and putting it into a new machine, it's never been used that way before. So we thought there was a bit of engineering risk there, so we could get to a terabit per day at the end of the seed round. So we'll be, with these milestones, we'll be going out and raising a new round of funding to up the throughput by about 1,000x, and our goal is to get to a petabit per day of writing speed, so 125 terabytes at that point in the next 2 or 3 years. And one of the goals in designing the terabit per day machine was to have a scalable design so that we could further increase the throughput given more funding and time. And so we've already begun sort of that process, and we think that there's a large market and demand for a very long-term, stable archival medium like this. But we also want to turn it into an active archive so that you can compute directly on those molecules without having to sequence it and pull it back out into a hotter tier of storage. So that we're kind of shifting the paradigm here a little bit, but the coldest tier of storage that you can imagine, DNA, could actually also become the hottest tier because you're computing directly on it without having to pull it back in and out of DNA.
Any other questions? Up here at the front, if one of the volunteers doesn't mind jumping up here. Thank you.
Thank you for this fascinating presentation, by the way. I'm very grateful to you both. I'm Sandra Schulberg from IndieCollect, and I'm fascinated by lexicons. You used the word a few minutes ago, and I'd like to know, at the moment, what is the size of your lexicon? And I believe you said it's the ones only. You're not worrying about the zeros. So, but you've abstracted to try to get more quickly and more efficiently to the end result. You've abstracted some number of terms, let's say. Could you tell us what number that is?
Yeah, so since our machine can only get to a terabit per day right now, that's really the practical size of the lexicon that we need. And so we've developed it up to that point, but it's a readily expandable lexicon where you add on additional layers of DNA molecules, and that can, in a very systematic way, exponentially increase the size of your lexicon. So in essence, you're just adding new letters of the alphabet each time you add a new molecule to the tail end of the growing DNA molecule.
And it is a little proprietary.
So in terms of the number of alphabets that we use on the order of about 100 different words that can combine into trillions of different or hundreds of different letters to combine into trillions of different words. I think that's the level that we can speak at right now.
All right, I think I saw one more hand, and I think we do have time for one more question. There in the back, if one of the mics can go there. Thank you.
So with the longevity of this material comes sort of a new challenge about how to speak to the future, 300, 500 years from now. Have you guys discussed plans for that? Currently, a lot of collections sit within libraries and other institutions that sort of carry along the information, the metadata that goes with that material. But with this, it sounds like there's some implications that people may not have addressed or thought about.
Do you want to talk about maybe ARC a little bit? I'm sure Nova would be okay with that. Just mentioning like what some people are thinking of doing. It doesn't have to be definitive, but.
Right, so there are people like the Internet Archive, ARC Mission, whose sole mission is to archive all of humanity's knowledge and keep it forever. I think what DNA is doing is solving one piece of a much larger problem. So with any media that you're storing information in for long periods, you have problems at the hardware level, and then you have problems at the software level. With DNA, I think what we can say is that the hardware problem is much less, because we'll always be able to read back the base pairs as long as our technological advancements don't go back, because we can already read DNA base pairs. But once you read those base pairs back, will you be able to translate that back into the original message? And will that file be in a format that's readable? I think those are problems that are in the software layer that are common to this industry, no matter what hardware you choose to go with. With DNA, you at least have the hardware taken care of. But with that in mind, we're actively looking at things like semantic metadata, so that you can search through things after the fact without knowing beforehand what you wanted to search through before. And the parallel computation that DNA-based storage enables actually allows you to store unstructured data and be able to search through them much more efficiently later on without having indexed it as extensively as you would on traditional media. You can actually do the searching and indexing at runtime. We're speaking with lots of archivists that are experts in this field, but right now we're not trying to invent anything new in terms of a format that'll last forever. I think that's a bigger problem than one startup can solve on its own.
Well, I'd like to thank Hyunjun Park very much for joining today.